Thank you very much for coming to this webinar about amlexinox, which is a repurposed drug that you'll be hearing from shortly from Dr. Bruce Bloom, the CEO of Fortuity Pharma. And you'll also be hearing from Dr. Kathryn Helde, who will talk about the genetics of Syngap and why and which mutations she thinks are amenable to this. My name is Mike Gralia, and thank you. I am just here to give you a bit of context on, next slide, please. The what and why for repurposed drugs. And this by itself could be like an hour-long talk, but I'm going to keep it much, much simpler. Before I jump in, I want to define repurposed drugs. Repurposed drug is a drug that we have already used in humans. We've already given it to lots of humans for some other disease. So the question of safety has been answered largely. And the question, and all of the drugs that are out there, we find a drug that does a thing and then we test it for that thing, but that drug could have other impacts. And if we can find a drug that in some way benefits our population, our precious Syngapians, we want to use it. And then next slide, please. As a reminder, Syngap was discovered in 1998. The first time we found it in humans was in 2009. What you're looking at is a timeline of every paper written about Syngap from 1998 to today. And those bars are the number of papers in each year. So you can see that things are really heating up and that SRF has played a role in that. Don't worry, I won't take you through this whole slide, but just on the far left is the paper where we found the gene. And then in 2009 is where we found it the first time in humans. That was 25 minus 9, 16 years ago. So even we are working at light speed to get therapies for our kids, but these things really take time. And if we really had time for me to walk you through the slide and explain all the achievements that I call out with these lines, we're moving incredibly fast, but we still don't have therapies to help our kids that are indicated for Syngap. Next slide, please. And as a reminder, we have a growing population of kids around the world. This is our quarterly census, which I think every Syngap family is familiar with. And every quarter the number goes up. And then really the reason I put this slide up is for a couple reasons. First, the number is growing. And even if we start with genetic therapies in a year, because best case scenario, Camp 4 would be starting a trial in a year, right? That is a trial. It'll be maybe 10 or 20 or 30 or 50 kids. And then there'll be another trial, which may be 50, 100, 250 kids. That we have already 1,500 kids. By then, we're going to have 2,000 kids found, maybe 3,000. So both in terms of numbers and in terms of geography, we have a lot of kids out there suffering from Syngap that do not have therapies, which is a case for repurposed drugs, right? If we can find a drug that is safe and effective and already made and already understood by the medical community around the world, maybe we can find drugs to help our kids now, while we're waiting for these very precise genetic therapies to be broadly available, right? So just a reminder, we have a lot of kids, we have a growing number of kids, and we have kids in countries around the world. Next slide, please. 2 points on this slide. First point, when SRF started, this slide was blank. This is the pipeline of SRD therapeutics. So this is every molecule that we are aware of and are allowed to talk to, because there might be something that we're aware of that we're not allowed to talk to. But if that was true, I couldn't talk about it, so I'm not. But this is every therapeutic or potential therapeutic we are aware of on the planet. And the purple box is small molecules. So this is drugs, either new drugs or repurposed drugs. So as in the case of like Rubor International for fenfluramine, that would be seeing if fenfluramine helps in Syngapians. Many of us are familiar with the work at Weill Cornell was Dr. Greenspan for Ravicti. This is a classic repurposed drug. Ravicti is for urea cycle disorder, and we're trying it for Syngapians. And then some of these other molecules are novel, and some of them like Chow and Rare Base are like nortriptyline and NAL and other repurposed molecules. So these are small molecules. This is the world we're in right now, as opposed to genetic therapies like ASOs and AAVs. And the important point here is every arrow under ASO and AAV is under preclinical. They are not yet in humans. And We are hopeful that maybe in a year we could be in humans, but as I just said, that's going to take time to get through trials. Whereas some of these purple lines are already in human. Why? Because they're safe. We already know that you can put them in humans and that it's worth the experiment. And so what we're talking about today is amlexinox, and could we put amlexinox on this slide with an arrow under inhuman? And that's the challenges and the opportunities is what we're going to talk about today. But this is what we're talking about: repurposing a small molecule that Dr. Bloom will introduce, um, and seeing specifically if it can help syngapians. And then do I have one more slide, Kathryn? Yes. And just as a reminder, because if you're a family, you've probably fundraised for SRF or donated to SRF. At the end of last year, we had spent $6.2 million on SYNGAP research. This is not overhead. This is not staff costs. This is just money we've given in grants. Of that $6.25 million, $1.65 million, that second column, was under our purpose program. And that is looking for and working with repurposed drugs. And through that work, we've still got a hit list from Unravel we're working through. We found NAL, we found nortriptyline. And what we're hearing about today is yet another drug that might be able to help our kids that's repurposed. And so far, we've spent nothing finding it. It's all been a fortuity and Dr. Bloom's good work. So it's a really exciting opportunity. And I hope you understand now why we're having this webinar, in part because we want to have all this information in one place. So thanks, Bruce, for doing this, but also because it's exciting to think that there's another molecule out there we could use to help our kids while we're waiting for precision therapies to come online. And with that, I want to hand it over to Bruce Bloom, who has been a leader in rare disease since the first day I started. And he has an incredible bio, which I will not spend our time going through, but and maybe if you don't mind, Bruce, you'll give some context. But Bruce has been generous with his time and his advice and his guidance for as long as SRF has existed, and we're very grateful. And I'm going to hand it over to you, Bruce.
Great, thanks. So, yeah, I met Mike at a rare boot camp a long time ago, and let's see, I don't know what's supposed to. Nothing's going to go up on the screen because I don't have any slides. So, Kathryn, that's fine, right? People can hear me and see me, whatever they need. Okay, great. So, yeah, I met Mike 5, 6, 7 years ago at a rare boot camp. I have been in the rare disease, drug repurposing, small molecule space since the year 2000, when I took the helm of a family foundation that we eventually turned into Cures Within Reach, which is a public charity that still exists, and it's one of the leaders around the globe in drug repurposing. And I've always believed that one of the great public health opportunities in the world is to find all the uses that you have with drugs that already exist that are safe for human use, and finding new purposes for them. But of course, the economic incentives to repurpose an inexpensive drug isn't big enough to get most pharmaceutical companies to work on it. I started Fortuity Pharma with a couple of other people. It's a passion project. It's a public benefit corporation, which means we can put the patients first, not the shareholders. And our goal is to bring out repurposed drugs at the lowest possible cost, at the soonest we can do it, to get things into humans. And the reason we're starting with this drug amlexinox is because 3 years ago, I met some parents at a different boot camp, and one of the parents had given his daughter, who was at the time 9 months and had a very severe phenotype of a seizure disorder called worry syndrome, WOREE. And at the time, she had no verbalization and no voluntary movement. And he looked online and saw preclinical evidence that this drug amlexinox, which at the time was for sale in Japan as an asthma drug, might allow read-through of nonsense mutations that create a premature termination codon as DNA is being transcribed into RNA, and then RNA translated into proteins. He gave his daughter this drug under the supervision of their neurologist, and within a week, she was moving and making noise, and she got better over a 4-month period of time. And then it turned out that Takeda, the maker of the drug, had stopped marketing it, and the supplies dried up and he couldn't get any more. And unfortunately, she went back to baseline and has been there ever since. And so I met them and said, we must be able to figure out some way to get this drug restarted. And so that's what Fortuity Pharma has been doing. So the name of the drug is Amlexinox. Its primary indications when it was approved in Japan in 1987 was as an asthma drug for chronic use. It was also approved in 1994 in the US as a molecule in an aphthous ulcer paste that was used in the mouth for canker sores. It was discontinued in the US in 2014, mostly because there were better aphthous ulcer pastes. And in 2019, it was discontinued in Japan because there were better molecules for asthma. It was not withdrawn from either market for safety issues. And in fact, as an oral drug, which is what it was in Japan, there's no documentation over more than 30 years of use of any significant side effects in the adult population. The only place it was approved as an oral drug was in Japan. It was used there for a long time. It was not approved or tested in the pediatric population. I've heard anecdotally it was used in the pediatric population, but it wasn't approved for that. In the US, it was for aphthous ulcers, also in the adult population. Again, I've heard through grapevine that it was used in children as well. The mechanism of action for its primary indication is it's very antihistaminic. So it targets two kinases, one called TBK1, which is also known as tank binding kinase 1, and the second one, IKK epsilon. And these are pro-inflammatory signaling pathways, and this blocks those pathways. It inhibits the release of histamine, and it downregulates the immune system cells that respond to chronic inflammation. And so it's a very effective, low-cost, non-steroidal anti-inflammatory. It's not a pain-relieving non-steroidal anti-inflammatory like aspirin or ibuprofen, but it is anti-inflammatory, and it also mediates it's a mediator anti-releaser due to its histamine inhibitory properties. That might be helpful in SYNGAP1. I don't know, but that's not the reason we're thinking about trying it in this patient population. The fact that it can read through nonsense mutations that lead to premature stops, what that means is as the DNA is getting read, there's an error in the code, and it tells the transcription process to stop short of making the full-length mRNA. So you end up with this small piece, which is degraded by a process called nonsense-mediated decay. Interestingly enough, this molecule in Amlexinox also inhibits nonsense-mediated decay. So in cellular models in over a dozen diseases, not SYNGAP, but in similar diseases, neurodevelopmental diseases, things like Charcot-Marie tooth, cystic fibrosis, Duchenne muscular dystrophy, epidermolysis bullosa, AGU, and a couple of others. They have done both cellular and animal studies that show that the molecule will read through, in other words, allow a full-length mRNA to be created from the DNA, even though there's still an error in the mRNA. And then it blocks the nonsense-mediated decay. So it doesn't break down that piece of mRNA, which then gets translated to a full-length protein, which still has potentially the incorrect protein where there was where the nonsense mutation was in the first place. But that full-length protein may be functional. And so for those individuals who have SYNGAP1, where their variant is caused by a nonsense mutation that leads to a premature stop, it's possible that this drug will allow read-through and production of a full-length protein. There is some evidence it crosses the blood-brain barrier. It is a small enough molecule with the right kind of polarity to cross the blood-brain barrier. We have not done any studies to show that it does, but in worry syndrome, the one patient that we know that took it, it needed to have crossed the blood-brain barrier in order to get the impact that it had. The dosages. So in Japan, it was given to adults as a 50 milligram tablet, and patients were advised to take it 2 or 3 times a day. And at that dosage, there were no side effects that were seen in that patient population. It has several other off-target effects. It seems to lower A1C in type 2 diabetics. And because of that, it helps with some weight loss. And there is a weight loss clinic in Denver, Colorado, that asked a compounding pharmacy in Kentucky to make this molecule. And it is being made at GMP grade, which means it's safe for human use. And we had it purity tested independently by this compounding pharmacy called TaylorMade Pharmacy. And it is available for purchase and prescription and purchase. And patients are taking it for weight loss. We've talked to the clinic in Denver and we've talked to the compounding pharmacy, and they're both supportive of us. If individuals and their physicians want to try and do an observational study using this molecule, it can be made available in 44 states in the US. There's no global availability of this anyplace else, and it is not an approved drug in the US. It was approved as an API in the aphthous ulcer paste, but it is not approved as an oral drug. But there's exceptions in the FDA rules for molecules that are safe, so this compounding pharmacy can make it and physicians can prescribe it. We're talking to a number of other rare diseases where nonsense mutations create a genotype that causes the phenotype of the disease. And we're hoping to gather some observational data of both efficacy and safety so that we can go raise some money to begin proper clinical trials. There's some work that needs to be done before we can file an initial new drug application with the FDA. We have some partners at Vanderbilt University who are eager to help us to do that. So we need to be able to raise some money. We're hoping to raise money through program-related investments, which are donations or investments that foundations can make, where it acts part as a grant and part as an investment, and they can get paid back some or all of their money, or even with a little interest. Our hope is to be able to do this so that we get approval in at least one disease, and then to do an ongoing basket trial, so many of the 6,000 plus rare diseases could participate, so we can see how widespread the impact of this could be. So let me stop there, and maybe Kathryn and Mike have other questions, or Lauren, and I can answer those. I'm available to answer questions from you or your physician. You can get to me through Kathryn or Mike or Lauren or anybody else at Syngap, and I'm happy if they make my contact information widely available so that people can get a hold of me. I'm not promoting this as a therapy. I'm promoting this as an opportunity, as Mike said, to see if there's another molecule that could be added to the therapeutic pipeline in SYNGAP and other rare diseases where a portion of the population that has the condition might get some benefit from taking this really safe drug. I took it myself for 90 days just to see what the impact was, and I could not find anything that it did for me, either positively or negatively, but I wouldn't have expected it to, because I don't have a nonsense mutation. And luckily for me, I don't have any mast cell conditions or histaminic allergies. So, but but no changes to anything else that I could tell. And I had several blood tests during that time for other reasons, and nothing was different. So, Kathryn, I'll turn it over to you.
So before I give it to Kathryn, who's going to go deep on the science, I just want to underscore a couple points. Bruce and Kathryn, please correct me if I've missed something here. So Bruce, what we've heard you say is that this drug was sold in Japan and used in Japan for asthma. So presumably thousands, if not hundreds of thousands of people have taken this drug over years, if not decades.
Correct.
And there were no adverse side effects. So like, and you took it for 90 days. And the punchline is that we believe this is a very safe drug that humans can tolerate, no problem.
Uh,
And even though it had all these NSAID and whatever impacts, you've identified through this wonderful story of the worry syndrome kid, and it might be great to get a link from you somewhere, Bruce. There's got to be a profile of this kid and the incredible, because
yeah.
worry syndrome for the audience who's all SYNGAP, not a good disease. And the fact that we were so quickly able to see a change in the phenotype, maybe Bruce, you can elaborate on that, tells you that, as Bruce said, it got to the brain and it did what we're talking about. And what it does is it ignores mutations in certain kinds of mutations, which Kathryn will talk about. And so that's not specific to the worry gene. That's any gene. If you have this specific kind of mutation that Kathryn will talk about, it could read through it, which means that for your Syngapian, if you have these kind of mutations, basically in the presence of this drug, the body could ignore the typo. And all of a sudden, you've all heard me say good copy, bad copy, one makes protein, one doesn't. That one that doesn't in the presence of this drug could start making protein, right? So
Yes.
it's pretty exciting. And so I just want to underscore, I'm going to ask you, Bruce, if you just spend one more minute talking about the worry syndrome, the phenotype, and what we saw. I want to underscore that this is a read-through drug. So if you have the kind of mutations Kathryn will talk about, this could basically make your good copy work. And then the last thing I want to emphasize is there is a GMP, which is the highest possible quality, source of this drug in the United States that you can legally secure with if your doctor is willing to write it. And that generally is like there needs to be a physician who's overseeing this. And what we hope with this webinar is that you, in conversation with your family, will say, hey, maybe what I would like us to see as a community for a couple of reasons. A, I want less SYNGAP kids to suffer. So there's a chance that this drug could help our kids the way we're about to hear about the worry syndrome kid. But B, the question that we all want to know is what happens when a Syngapian, a human, because we've seen it in mice 1,000 times, what happens in a human when you turn Syngap back on? We don't know that yet. And this could actually help us see that if it works as it did in the worry syndrome kid. So I'm super excited. And then Bruce, can you just talk about the worry before and after?
Yeah, sure. So this little girl is born and diagnosed with worry syndrome. She has seizures all the time. She's basically in a vegetative state. She has a feeding tube. She's making no noise. She's not moving, and her parents are obviously desperate. And there's really nothing, they were among the many parents who went to see their neurologist and other doctors and were told there's really nothing we can do for you. We can give her anti-seizure medications, but they didn't seem to make any difference. And the dad found the literature. And by the way, it's all up on the fortuitypharma.com website. You guys can, put a link to that in here. All of the preclinical research is linked in there. All of the old information that we have on mLexinox from Japan, all linked in there. So for your families and the physicians of your families, there's lots of information available in that space. So anyways, he gives his daughter this drug through her feeding tube, 50 milligrams in the morning, 25 milligrams midday, and another 50 milligrams at night. And you can go up, there's a link on our website to the Worry Syndrome Foundation website where you can see pictures and videos of Lucia. And the family's very open about this, and they're happy to talk to anybody about this. But For 4 months, every day she got 125 milligrams of this drug, and she continued to improve. Now, she didn't get anywhere near normal. She has a very serious phenotype of a multi-system condition, but she had a significant improvement, reduction in her seizures, improvement in her vocalization. They saw no evidence that she was having any untoward side effects on this. Now, obviously, she was 9 months when they started and 13 months when they finished, and so you're not really sure. But no overt signs, no. I mean, we know when our kids are in pain, even if they don't say it. We know when they're uncomfortable. We can see their feeding habits and their digestive habits, and nothing changed in this other than her phenotype improved. And then when the drug was withdrawn because he ran out of the last bit of dosing, she quickly went back down to baseline. So our belief is that she was producing enough protein to start to change the course of her condition. And then at the end, it was withdrawn, and she was now not producing protein, and the condition took back over. One thing I do want to say, and Mike, you and Kathryn and I have talked about this, and it's really important. We really think that we have to be careful about giving this to the youngest children with any of these diseases that aren't. Well, I'll explain it and then I'll tell the exception. So, nonsense mediated decay, which is part of what amblyexanox inhibits, is a very important part of the human, because you don't want bad proteins getting produced. And so it starts when kids are at their very youngest. When they're born, their nonsense mediated decay works. Interesting, there's some research that says nonsense mediated decay as a normal process is involved in the shaping of synapses and other neural connections in the developing brain in the youngest kids. And we know that you need to strengthen and prune those. There's so many. When a baby is born, there's way more synaptic connections than need to be there. And the process of learning and everything else prunes those down to the normal amount that is healthy for a human. And nonsense mediated decay actually plays a part of that process. So if you inhibit nonsense mediated decay in a newborn, you might change how that synaptic pruning and the neurological development happens. And so we think for all but the most severe seizure disorders, where the damage to the brain from the seizures is definitely going to be much worse than the damage from inhibiting some of the nonsense mediated decay, you might want to wait until a child is, and again, it depends on the severity of the condition and it depends on the disease, but you might want to wait till age 3, 5, 8, 13. It all depends. I sent Mike and Kathryn an evaluation that we did on a number of diseases, including SYNGAP, and each one, it looked like a different age was the right age to start. But for all of them, it was 13 and over. We think by that time, the brain is almost fully developed and you're not going to get much synaptic pruning anymore. Did I answer your question?
You definitely did. And I take your point. The challenge with Syngapians is half of the synapses that are being formed are defective because they don't have enough Syngap in them.
Right.
So it is tricky at the age you start. But if you've got a kid who's 7, 8, 9, 10, thereabouts, if you have the right mutation, it's tempting. So let's let Kathryn go from here. And I just want to quickly, and then I promise Kathryn, I'll stop. I just want to share your site, Bruce, because I was looking while you were talking, and to find that article you talked about for everybody to see it, I had to go to news and events. You guys can see my screen, right?
Yep.
I had to go to news and events, and I scrolled down and I couldn't find it until I hit view all. I had to click load more. And then when I load more, you get a nice article about amlexanox down here that your doctor might like. And here is the journey of hope for Lucia's story, Bruce. Is that correct? That's the one you're pointing people to.
Yep.
Great. Okay. Thank you so much again for all your work to
Sure.
help so many kids with this drug. Kathryn, what mutations with Syngap would this help?
Oh, wow. What a great set of talks from Mike and Bruce. I'm really excited about Lexinox as a potential drug for SYNGAP related disorders. And so now we're at the point in the webinar where I'm going to talk about and answer the question, which SYNGAP1 variants in our community, in our population, which are best suited to respond to a read-through drug like Lexinox. And the answer is going to be in the genetic variants and in the DNA around each genetic variant. So the genetic variant is what you'll find on your genetic report. And the DNA around each genetic variant is not on your report, but it can be found on the reference sequence. And there's a bunch of places to find that. And I don't expect anyone to do that, but I like doing it myself. So that's what this part will be about, is about the genetic variants and the DNA around it. Okay, so instead of making you wait till the very end, I'm going to give you the answer first, and then I'm going to show you how I got there. So this table shows you 6 different variants. They're all nonsense. 5 of them are arginine nonsense, one's a serine nonsense. And they all share the fact that they have a sequence, there's a sequence around them that is predicted to read through the aberrant, the mutant nonsense. Okay, so a few things to note about this table. I made this table as of June 18th, 2025. This is not a complete list of everything in the SYNGAP gene that might work. In fact, this came from the last Citizen Health data set that we got back in April of 2024. And if you have something that you think might be a good variant and you want me to check it out, please email me cso@curesyngap1.org. Okay, so this is the table that I came up with, and now let's talk about how I got there. So really the question is, which variants promote read-through? And like we said, Lexinox is a drug that we think has a high likelihood of promoting nonsense read-through, and that it might be helpful in SYNGAP. So I'm going to go through 6 sections. I'm going to try to do it quickly and without too much extra information. Number 1, what is nonsense read-through? Number 2, what are the DNA sequence rules for read-through? Number 3, what are the SYNGAP1 related disorder genetic variants in general? Number 4, what are the SYNGAP1 variants that fit the sequence context rule? So that'll be how I get to that table that I just showed you. And we could stop there, but I thought I'd go through number 5, a couple of my thoughts on the drug mechanism, and 6 references and further reading. Okay, so we're going to start with what is nonsense read-through. So there's only 4 words in that question, and 2 of them I think we need to define. So nonsense is a type of mutation, and at the DNA level, it's where one letter changes into another letter, and the effect on protein is that there's a premature stop. And so because of that, the SYNGAP1 protein is too short to function, may even get cleaned up and not even be around. Okay, so that's what nonsense is. It's a type of mutation where you have a single letter change ending in a stop. What is read-through? Read-through is a process, and read-through is a process where even though there is a premature stop, a full-length protein is made. How does that happen? It happens because an amino acid can be added instead of the process stopping. Okay, so that's what nonsense read-through is where you have a type of mutation called a nonsense, and there's a process that makes the problem kind of a moot point. Okay. So in order to go a little deeper in this, we're going to need to know some basic genetics. So how does the RNA encode proteins? For DNA, there are 4 letters, A, C, G, and T. They make up 64 unique 3-letter words. RNA is almost exactly the same. It has A, C, and G. Instead of a T, it has a U, just a chemical change, very similar chemical. And again, that makes the same 64 unique 3-letter words. So 61 out of those 64 words code for one of the 20 amino acids. The others, out of the other 3 of the 64, all 3 of those mean stop. So the words that mean stop are TGA, TAG, and TAA. So we'll hear that again later. Okay, so how does DNA encode proteins? This is a picture of that. Again, like I said, there's 4 different letters, A, C, G, and T, and they make 3-letter words. So what are the 3-letter words? They'd be things like AAC, AAA, ACA, and on, all the combinations you could make with 4 different letters and 3 different positions. So here in this top ribbon with the dotted lines around the box, this is what DNA sequence looks like. Honestly, it's kind of hard to see, right? Like you look at it and it's like it's hard to even imagine what that might say. So I decided to chunk it up into 3-letter words and show you what those 3-letter words might mean. So the sequence down here in color is the exact same as above. It's easier to see. It's got the same spacing and everything. I just put color around it so that you can see them in sets of 3. And then above that, I show what the protein amino acid is that this word means. So a CGA, that means arginine. A TCA, that means serine. And then if we go to the end and this orange, the TAG, that one means stop or termination, or if it's in the 1-letter code, it would be X. Okay, so this is the basics of how DNA encodes proteins. So now we're going to talk about types of mutation. So the first kind of mutation we're going to talk about is when there's a single letter change. In this case, we're going to talk about C to T. So this is the same, this is the same as the slide before. So I've got a CGA here that means arginine, a TCA that means serine, and so on. I've got 6 different words here. That's where you can see that in these numbers 1, 2, 3, 4, 5, 6. And that would be akin to the P address. Above that, I have these numbers 1, 2, 3, 4, 5, 6, 7, 8, 9. It's almost like a waltz, and that's going to be the C address. That's going to be similar to that. So if we have a C to T, the CGA, which encodes an arginine, is going to become a TGA, and that TGA doesn't encode arginine. That actually encodes a stop. Similarly, if over here at C address 14 and the P address 5, the C becomes a T. Now instead of a GCT encoding alanine, you have a GTT, and that encodes a valine. So these are 2 different types of changes. In both cases, it's a C to T. And depending on the sequence around that C, that, this initial C, that is what leads to whether it's going to be a missense or a nonsense. So when a single letter change turns an amino acid into a stop, that's a nonsense. That's here in blue. When a single letter change turns an amino acid into a different amino acid, that's a missense. That's here in green. And how do you know which it's going to be? It's entirely dependent on the sequence around the mutation. Okay. So next, let's talk about adding or removing DNA. If you add or remove DNA, you can cause a bigger kind of problem called a frame shift. So I'm gonna show you what that means in 2 examples. First, I'm going to show you what happens if you remove 1 letter and what the result is. So let's say that there's a mutation where this red lightning strike is on this first letter, which is a T in the serine codon. So instead of TCA, we're going to remove the T all the way. So what happens when you remove 1 letter? Well, you can see that on this line below. So what happens is the CA move over. So it's going to go CGA, the T's gone. So you start with CGA, the T's gone. So it goes CA, and then the A from the next codon becomes the last letter of this 3-letter word. Okay. So all these diagonal lines show you that when you pull this out, the A and the C are covalently attached, and it's just like a string. You just pull it over. Okay. So instead of being in frame 1 here, where you have ARG, SEER, isoleucine, ASP, you still start with ARG in frame 1, but as soon as you get this mutation, it shifts, and now it's in a different reading frame. It's going to be in reading frame 2. And what you get is, instead of serine, you get a glutamine. Instead of isoleucine, you get a serine. Instead of ASP, you get a methionine, and so on. So all these things in brown down here, these are not the right letters. So this is not going to work. What happens when you remove 2? So now instead of just removing the T out of the serine, we're going to remove the TC. So these lines show you how this shifts over. Now we're not in the second reading frame. Now we're in the third reading frame. So instead of CGA TCA, we've got CGA AAT. And so again, I've put in brown all the different amino acids. And honestly, they're all different than the ones above that they're supposed to be. So again, I just want to show you all 3 reading frames. So now at the top, we have reading frame 1. In the middle, we have reading frame 2. And you can see how these are hybrid with this coloring. You can see how it's hybrid from the sequence above. And the same in reading frame 3. It's also a hybrid. It's just scooched over one more. Okay. So now instead of showing you just in a short sequence, I wanted to show you how this looks in a gene. So this is a screenshot from the genome browser. This is the end of an exon in SYNGAP1. And what I want to, with this next set of slides, what I'm going to convince you of is that the DNA in the exons, which is going to be the DNA that would code for all these blue things, all these blue amino acids here, the DNA in the exons codes protein in one reading frame. It doesn't code protein in all 3. There's one reading frame that's the right reading frame. And that the non-coding DNA is full of stops. So the non-coding DNA includes the intron here. So the intron is going to be full of stops. And then in the exon, the 2 frames that aren't the reading frame, those are also full of stops. So I don't expect you to believe me right yet, but I'm going to show you how this works. So again, the main points here are that the right frame, which is the reading frame for the gene, that has no stops in it. And that the wrong frames all have stops. So this is the same sequence that we had on the previous slide. I've just added all 3 reading frames above it. So you can see this starts QGFLS, this top line here, QGFLS. This is the same open reading frame all along here. There's no stop here. Now, once we get into the intron, yes, there is a stop in frame 1, but this is after we cross over from the exon to the intron. What does the stop look like? The stop is this red box with the white asterisks in it. So there's a bunch of them. There's a stop in every single frame in the intron. And then there are stops in frame 2. There's stops in frame 3 in the exon. Okay, so read-through, we're looking at a read-through drug. Read-through requires the right frame to be preserved and within an exon. So if you preserve the frame here, but you go into intronic sequence, that's not going to work. So if you read through this intronic sequence, that's not helping you make a good protein. It only works if you're in the right frame in the exon. Okay, so I've gone over a bunch of things. I want to show you some sample changes and their consequences. So over here in blue on this table, we're looking at everything that is tested and reported. So the DNA change is what they test for and report. The location of the DNA change is what they test for and report. Okay, so first 1, 2, 3, 4. The first 4 rows here are all single nucleotide changes, so either G to A or G to T. So if you have a G to A, that changes one amino acid to another, that's called a missense. If you have a single nucleotide change that changes an amino acid into a stop, that's called a nonsense. If you have a single nucleotide change that has a minus or a plus after the first number, that is going to be in an intron. And so those are called splice variants, intronic variants or splice variants. And finally, if you have an insertion, a deletion, or a duplication of information that shifts it out of frame, so that would be basically if it's not divisible by 3. So if you don't take out a whole amino acid or a number of whole amino acids, then you shift frame. And so if you delete a G or a GA, or so 1 or 2, or if you insert 1 or 2, you're going to get a frame shift. Okay. So I want to show you a little bit more about this. Out of all these types of variants, which ones are protein truncating variants? Missense, no, it's not a protein truncating variant. Nonsense, intronic, and frame shift, yes, those are the ways you could, those are the ways you can get a protein truncating variant. I just went ahead too fast, but I'm gonna go with it. So what's new about this slide is I added in here in purple what the read-through requirements are. So you can see that at the top. What are the read-through requirements? They're here in this purple line. So the read-through or the variants that will work well for a read-through drug, they need to have everything here in purple. So it needs to be a single nucleotide polymorphism. It has to be one letter change. It cannot be this deletion or insertion. The location of the DNA change needs to be in an exon. It won't work if it's in an intron. The protein change, it needs to be a stop. And so if you're looking at your genetic report, it would end in a star or an X or a TER for termination. The type of variant is going to be called a nonsense. And so, yes, it's a protein truncating variant. Yes, it's in an exon, and no, it doesn't have a frame shift. So the only type of mutation that satisfies all those things is the nonsense. None of these other types satisfy that. Okay, so I hope I have explained to you why you need a nonsense. The nonsenses are the only ones that satisfy the requirements for what you need to get read-through. The next thing we'll talk about is the rules for read-through. So this is not something I'm sort of knew about naturally, but Bruce was kind enough to send me 3 papers that really explain the sequence context for read-through and what makes read-through work better. Now, I'm not going to go through all the tables, but one of the papers had a lot of really easy to understand information, and it looks sort of like this. So if there's a bunch of tables, there's a bunch of drugs here that they're looking at, and they test a bunch of things, and so they look at the 3 different words that mean stop, and they look at the sequence context around that. And so this is the kind of data that I used to write down these rules. So from the papers, what we found is 2 main things, one in each of these boxes. The most important is at the top, and that's that the TGA codon is more likely to read through than TAG, and that's more likely to read through than TAA. So TAA has the tightest regulation. It's more rare for a protein to continue on after a TAA. The TGA is the most leaky. It has the sort of loosest regulation. So I thought that for a small pilot in our community, that starting with TGA would be the best idea. Now, in addition to the TGA, there is some other sequence context. And so I have this down here. The ideal sequence context is, it starts with a C in this +4 position. A C confers a big advantage. A T confers a little advantage. And then in addition to that +4 position, there are 3 other positions where if you have a T here, it does a little better. If you have an A at +5 or an A at +9, it does a little better. I've written all the other positions as N's, meaning it could be anything. So this is the ideal sequence to promote read-through. Okay, so now we know what the rules are. Let's talk about SYNGAP1 variants. I'm first going to talk about them generally, and then I'll talk about how they fit in the sequence context rules. So in general, I have a graphic here that shows you about how many variants there are of each type in our citizen health cohort. So this is 189, 190 patients that are in the citizen health cohort, and 81.5% of them are protein truncating variants, and 12% are missense, 6.5% other, and then out of these PTVs, 32%, 41, and 8.5% are nonsense, frame shift, and intronic. So when we're looking at, so how many nonsense are there? 32% of the whole thing, 32% of the whole population is nonsense. That doesn't mean 32% are going to have the good context rules or even have a TGA, but that's what we're looking at. So we're already looking at a third to start. Here is a list of all the nonsense variants that we have in Citizen. So on this table, it's showing you in the left here, I have written the number of patients that are in Citizen. And then on the right, and these are all nonsense. See, every single one's a nonsense. And then we have the C address and the P address. So this represents, on this table, 32% of the total patients, 22.5% of the total variants. So how are we going to figure out on here who's a TGA? It doesn't say on the genetic report. So I had to think about how to figure that out. And that's what I'm going to talk to you about next. So what can turn into a TGA stop codon with one change? So if we're trying to find a TGA, here's a CGA in red here. If the C in the CGA turns into a T, what do you get? You get TGA. Similarly, if you have a TCA and you turn this C into a G in this green circle here, what do you get? You get a TGA, right? So there's a bunch of ways you can get a TGA. This table shows you all of them. And so if you're looking at if TGA is what you want to end up with, and you want to get there with only one nucleotide change, you could change the first position, right? So the first 3 lines in this table are the 3 different things where the first position could change into a T. If it's changing into a T, that means it can't be a T. So it has to start out as not a T. So the 3 things that aren't T, A, C, and G. So here's how that would work. Similarly for the second and the third positions. Okay, so if you start with a CGA and do a C to T, you get the TGA. So CGA is an arginine. So now we know what to look for on the genetic report. I look for a C to T in the C address and an arginine that's becoming a stop. So this table is telling me what I look for. Similarly, I could look for a G to T as the change with a glycine becoming a stop. Okay, so I'm not going to do the rest of the table, but I think you're getting it. This is how I figured out what to look for in the genetic report. Because all we have is the genetic report, right? We have to figure out what it does say that can help us. This is what we look for. Again, there were 59 patients that had nonsense variants in the first cohort. Those 59 patients are spread out over 34 different nonsense variants, and a lot of them are arginines. So you see up here, arginine is one of the ones we're going to look for. A bunch of them are arginines. So there's actually 10 different arginine variants that represent 34 patients. Okay, so this is that same table from before, but now every time the line is colored, that means it fit one of the it fit one of the rules from the previous table. Okay, so now we know these guys are TGA that are in color. All the ones that are still in white, they do not have a TGA. They either have a TAG or they have a TAA. And so we're not going to start with those. Right. So now I've got this set of TGAs, right? So now I've got the SYNGAP1 TGA nonsense variants here on this table. And now I have to look at the sequence context. Again, this is not on the genetic report. Where is it? It's in the reference sequence. So now I go to the reference sequence. First thing I do is I look up SYNGAP1 C403. When I do that, I get to this C right here, this C. And since I know there's an arginine and I see it's a CGA, I think, yay, great, I'm in the right spot. And so I look to the left and I see an AGC and I write that in the table. I look to the right and I see a CGG and I write that in the table. So that's the first step, is I look each of these up and I write down the sequence on either side. The next thing I do is I go through and I say, okay, what's the sequence context? The T, is there a T in this position? So I go down the column and 1, 2, 3 of these have a T in this position. I look in the +4 position. 3 of them have a C, none of them have a T. I look in the +5 position, 1 has an A. For the purposes of this chart, I did not look at the +9 position, partly because we're about to get another data dump from Citizen with 220, so I'm going to do even more. So I didn't want to spend too much time on it, but this is what I got so far. So now when you look across every single one of these, I'm showing you here the reference sequence, but every single one of these after they have this change, all of these will be TGAs. Okay, so down this middle column here, these are all actually TGAs for the patient. And at least for their mutant allele anyway. And so all I can do is check out which ones have the best context. So the C was the most important. So we're going to start with those. And of the ones with C, only one of them has another additional context that matches. So that's why this green one, this P0817, that one becomes the one I call like the highest, that has the highest predicted response. These ones that are called better, they have the C, and then these ones called good, they don't have this, they don't have the +4 position, but they have an extra context position, the -3 or the +5. Okay, so now you see how I've predicted response and done this sort of qualitative thing. So let's take away everything that's in white and just look at the ones that had the better sequence. Okay, so now we're down to our smaller table. This has 6 different nonsenses on it. They all have some extra sequence context, which I've shown you here. There are 2 more things on this table that I want to talk about. 3 actually, 3 more things on this table. One is one is the position within the gene. So there is evidence that if you're closer to the 5' end of the gene, say 10 or 12% of the way through, you're only 1/10 of the way through the gene, that the read-through may not work as well. Also, if you're farther down the gene, it may work better. So it's possible that the position effect here may affect things. So that has to do with how well will it read through. The second thing doesn't have to do with read through. It has to do with after it reads through, how well will the protein work? So if in each of these cases an arginine is put in or a serine for this guy, then you just have the wild type protein. That's great. But what if something else is put in? If something else is put in, that means it's going to be basically a missense. It's going to go arginine. It's going to go arginine to a new amino acid. And so I decided to just point out which domains these are in case that has an effect. So again, this is not something I know a lot about. This is not something we can really predict. This is something that we can check out, and we can look at basically whether it works or not, and try to think about that afterwards. The third thing I wanted to talk about is population variation. So when I want to point out again, this is all this sequence in the middle. It's all based on reference sequence. This is not patient sequence. I don't know what the patients really have here. But since I don't know what they really have here, I looked at the EPI25, which is a broad data set for population variance. And I looked for each of these codons. I did not see any variation in the population at all on either side of each of these codons. So that made me feel pretty good. It made me feel like, okay, this is probably the sequence that we see there. Okay, so once again, here we are with the chart I came up with. The only thing I want to say here is to add on that there's different ways to look at how many people this is. There's 27 patients on this chart. Not all of them are going to be the right age. Not all of them are going to want to do this. That's not what this is about. It's more about how does this cover the population? So I was sort of surprised to see that over 6 variants, that's actually 4% of the population. 14% of the population. This also is 46% of the nonsense population. A different way to think about it is by variant. And so there are 6 out of the 147 variants in this cohort. That's 4% of the variants. And if you look just at the nonsense, it's 18% of the nonsense variants. So if you're trying to understand how likely was it for you to be on this table, these are the numbers I came up with to kind of explain that. Okay. So we're done with what are the variants that I know of that currently fit the sequence context rules. So I'm going to go for a couple minutes on my thoughts on drug mechanism. So one thing that Bruce has told us, or has told me, and is going to tell us again, is that not only does amlexinox work for read-through, it also inhibits nonsense-mediated decay. So nonsense-mediated decay, or NMD, is where the mRNA will be basically chopped up and recycled. So if you're getting rid of the RNA, that's going to reduce the number of transcripts available to even do the read-through, right? So if you have a high level of nonsense-mediated decay, it's going to be hard to read through anything because there won't even be that many transcripts to read through. So it's important to know that amlexinox inhibits nonsense-mediated decay, and therefore there will be more chances for the mutant mRNA to be read through. And as I just told you about, the tolerance for whatever is inserted will be variable based on position. Some places in the gene will do better than others when you have a wildcard amino acid. Okay. So in order to talk about drug action, I want to go over what the components are that are needed for protein production. So I'm going to tell you about protein production, and then we'll look a little bit at where we think the drug might act. So in order to create a protein, you need 4 things. You need the ribosome, which is this big dark gray cloud here. It's made out of proteins and ribosomal RNA. You can see a large subunit, you can see a small subunit. Second thing you need is an mRNA. These are the instructions for a new protein. They're nucleic acid. It has a starting end, which is 5'. It has an ending end, which is 3'. It has a bunch of nucleotides, and it has 3 positions in it where the action happens. in addition to the ribosome and the mRNA, which are shown down here in the middle, you need all this stuff in the gray box on the right. So in the gray box on the right, you can see these colorful tRNAs. And the tRNAs are these ovals. So there's a purple oval, a green oval, a turquoise oval, and a red oval. So you need tRNAs that are charged with amino acids. The amino acids here are the purple star, the green star, and the blue star. You also need the stop tRNAs, and that's what this red one is. And it doesn't have an amino acid on it, but it does recognize termination factors, and that's what these big red things are here. So if you have all four of these things in the cell, you can, these are the things you have to have in order to make RNA. There's a 3-minute video on translation basics here, and I encourage everybody to look at this. It's actually fascinating. It's a nice, it doesn't cover stop, but it's actually really cool to see. Okay, so this slide looks confusing, but it's really the slide before. So you've got the big black cloud here, the gray cloud of the ribosome and the silver, the gray ribbon of mRNA, right? You've got the tRNAs on the side that are coming in. And now we're going to talk about the 3 positions. So in blue here, the A site, that's where the tRNA has to find its match with this site here on the mRNA. That's the action site. We're going to come back to that for drug effect. In orange, the second site is called the P site. That is where we're going to link this orange amino acid to the green one before it. So that's where the growing protein is covalently created with one amino acid to another. And then the green site here is the E site. That's the eject site. So this green one used to be in the P site and had the methionine. And then once the next amino acid once it moves over, the next amino acid can get on. Okay, so the E site is where it's going to eject. So this is going to go out and be an empty tRNA. These empty tRNAs have to go get charged with amino acids to start over here again. Okay, so you see these arrows, you see the stuff coming in and going out, and this is going to grow longer and longer, and this is going to ratchet. It's going to ratchet across and go down the whole length of the mRNA. And I should have shown this ribbon extending really far and looping all around and being really long. So what is happening over here in blue? Well, there's different concentrations of the tRNAs that are charged with amino acids. And then there's competition. There's competition for them getting into this site. So what do we think Lexinox is doing as a drug? I think it's probably affecting the timing, whether this is going to be a go or a pause. And I think it's could be affecting the affinity. So the affinity is how this tRNA is going to recognize the RNA site here. Not only can these 3 find each other and match up, but also it can also touch this one here. So if you remember, if this is let's say this was the stop site here, the CGA became a TGA, the next, if this was a C right next to it, that would have the best chance of read through. Okay, so that's pretty clear. Also, this site here is the -3, and then there's some more sites over here. Okay, so we think that what's happening is the drug is affecting the timing and the affinity of this stuff with concentration and competition that's making the protein. Okay, last slide is references and further reading. Please get in touch with us. Here's Mike Gralia's information, Dr. Bruce Bloom's information. Here's my information. Bruce is going to be the best person to talk to about Lexinox and repurposed drugs. I'll be the best person to talk to about SYNGAP1 related disorder and questions about genetics. If you want to look at the fertility website or the tailor-made compounding website, these are places you can go for stuff Bruce talked about. And then the 3 read-through papers are referenced here. Thank you very much. And let's do the Q&A.
Actually, Kathryn, I wanted to say, because you mentioned something just a second ago, the current drug is available as 40 milligram capsules. They're fairly small capsules, but they can be opened, and I have tried it. The molecule itself completely tasteless.
Great.
It's also insoluble in liquid, so it's a really nice thing to just mix into applesauce, and it can also be washed down a feeding tube. So there's lots of ways you could think of taking this if you were an individual, especially if somebody doesn't swallow meds really easy. There's still ways to get the drug.
Yeah. I might have misheard you. Did you say it's soluble or insoluble?
It's insoluble, but it mixes really well. Like I've taken.
Okay, so slurry. It could
it.
make a slurry or something.
Yeah, you can put it in applesauce and it just mixes in.
Okay.
It doesn't. It's a very fine powder, so it doesn't taste grainy. It won't change the taste of the applesauce at all to somebody who is reluctant to or unable to swallow a capsule. And I think your point about how much protein could be made. You went through all of the reasons why some read-through is easier than other read-through. It's also important to note that depending on where the variant is in your sequence and which parts of the final protein are critical for function, you could have a lot of read-through and a non-functional protein simply because of the error where the amino acid that's getting replaced. But you could also have a small amount of read-through that creates a very functional protein. So I think we're not going to know until we see what happens in individuals when we do this. And we certainly could go back and test it in individual cell lines and see what happens in the cells themselves. I don't know, to Syngap, do you have a nonsense animal model of any kind where this could be tested?
Yeah. Not in arginine. It doesn't need to be arginine. Arginine's just serine and glycine work. We have the Q503 mouse.
Yeah. So, maybe.
Oh, wait a minute. You're right. Actually, I have to make sure that's a TGA. I'm not sure. Yeah, it has to be a TGA. So I should look at that. Let me see. I'm going to look at that really fast.
I think the other thing to think about is even though the other stop codons don't read through as well, we don't really know how much SYNGAP is required to change the phenotype. So if it does work for the one that is the easiest read through, obviously, as Kathryn said, then we might want to expand the observational study. And the last thing that I'm thinking about here is if any of the physicians want to talk to, if we want to have a meeting with any of the physicians, we're happy to talk about that and talk with them, because I don't think read through is the kind of thing that most of the neurologists or other physicians are used to thinking about. It's been approved as the Atalurin was approved in Duchenne muscular dystrophy. It didn't really work very well, partly because I think the nonsense mediated decay was breaking down the fuller length mRNA. There have been some articles that are published where Atalurin and amlexinox were given in a cell line or in an animal together, because the amlexinox would also do read through and would inhibit the nonsense mediated decay. And in some cell lines, the combination was way better than Atalurin alone. In others, it wasn't. And so I think it depends on, like Kathryn said, all of the sequence of. And the other thing, Kathryn, did you mention this, that where the variant is closer to the 3 or 5 prime end, also, the closer it is, I think, to the 5 prime, the less likely it is to have a read through that's as successful as if it's someplace else on the gene.
Yeah, no, I didn't talk about that. There are quite a few things that are that could affect this, and since what we have is a patient population with different levels of risk tolerance, I think that's actually the more important thing. Also, I think what you mentioned, I didn't go over at all, which is sequence context tells you how likely how likely somebody else expects it to read through, that what it puts in, what it substitutes for the stop is not exactly clear. And your point about there are individual amino acids for which you're never going to tolerate, you're not going to tolerate the things that are put in instead. And so it's kind of like putting a wild card in, right? So read through is going to be like putting a wild card amino acid. We're not exactly sure exactly what's going to go in there. So if there's a tight restriction on what can function there, it might not work as well. So that's not that the read through didn't work. That's that the protein didn't work after the read through got in. So anyway, all I can say is you just got to try. I mean, you got to try it. For some, it's going to work. And I mean, in my opinion, some will work and some won't. And there'll be many reasons why it might not, but we can't sit here and figure that out. We just can't. So if somebody's willing to try it, they can tell us if it worked.
But we can't figure it out. So thank you. Amazing analysis. And I think everybody who watches this should appreciate that SRF is lucky to have someone of your caliber willing to go so deep on all those little letters.
Yep.
We can't figure it out, but we have done our best to identify the mutations where we think there is the greatest likelihood for success for a drug that we believe is completely safe in humans, for a drug that could make the mutant allele do something. I love and people who know both of us will be laughing because they're like, Mike's like big picture and Kathryn's like 18 levels of detail, to the point where, Kathryn's like, maybe it's just 2 figures. I'm like, it's kind of a cartoon. But I think the message I want patients to land with is, we have been talking to Bruce and aware of this for some time. We have thought about it. We've seen the worry syndrome girl significantly be affected by this. I want as many of our kids who can benefit to benefit. So the way we're doing that is we're putting this online. We're sharing. We've told you everything we know. We've given you a healthy dose of science. We've pointed you to online resources. The only thing stopping us from getting a kid on this is finding one of the kids with one of those mutations. And those mutations will be listed in the show notes and listed in the blog that we'll write. Have one of their doctors say, sure, it's safe, let's try it. If you can get a doctor to say, sure, it's safe, let's try it, I think we would see some significant change, likely pretty quickly. Personal opinion. And so I just want to encourage families, especially those who have that mutation. And then other families will be like, well, I don't have this mutation. To be crystal clear, this is only working for nonsense mutants. So if you don't have an asterisk or a X or a TER for terminate at the end of your P address, this isn't even in play for you. But if you have a nonsense, specifically one of the nonsenses noted, strongly encourage to take this presentation to your doctor and say, doctor, this is real science. Let's try it.
And can I, a couple other things, the drugs available, it costs about $125 a month to buy it retail from the compounding pharmacy if you're going to take 3 40-milligram capsules a day. If it was, when I started taking it, I took 1 a day for a week. Saw no difference. I took a second one a day, so I was then taking 2 a day for a week. I didn't see any difference. I was taking 3 a day, and I didn't see any difference. So, I ramped it up just to make sure there wasn't anything going on for me. But the little girl was on basically, about the same amount in her little body. That would be 120 milligrams a day, and adults were taking 150 milligrams a day. So I don't know that it varies based on body weight or anything like that. Your physician can figure that out with you, but I don't think there's, any concern about that. And I do think there are some observable changes that would be seen fairly quickly if you're producing protein in the right amount. So, I'm hopeful that it could be helpful, and we'll see, we'll see what happens.
Yeah, and I want to point out, if you're family with a genetics and you're not sure, please email me. It's easy email, cso@syngap1.org.
Julie Miles asked in the Q&A, and if anyone wants to talk live, just raise your hand. But Bruce, where's the compounding pharmacy, and will they mail it to us?
So the compounding pharmacy is in Kentucky, and they have authority to mail to 44 of the 50 states. It's the name of the compounding pharmacy is TaylorMade, TAYLOR, new word MADE, TaylorMade Compounding Pharmacy. You can go up on their website and you can see which states they mail to. For instances that we're working where somebody lives in a state where they can't mail it, they can mail it to a parent who can then send it on. And by the way, I've gotten some that I've sent on to Australia. Australia allows family and friends to send drugs over, through the mail, although they don't to England and a lot of European countries. There's a restriction. So I think we can pretty much get it anywhere we need to through one way or another. And the people at TaylorMade have been very, they're supportive of us. They said, we can't send it where we're not allowed, but we can help you figure out how you can get it there. So, and it just comes in a little bottle. They send you either a 30-day or a 90-day supply. Takes about 2 weeks, I would say, between 2 and 2.5 weeks from the day you finally get the prescription. And I think it would be easier if your physician worked with the weight loss clinic in Denver, because they already have an established relationship with compounding pharmacy. So they'll write the first prescription. After that, you just renew it online. So you don't have, once you get started on it, there's no hassle about continuing it.
So just to clarify that, if the Denver weight loss clinic writes the script and the Kentucky compounding pharmacy is filling the script, why then do we need our own doctors involved?
Well, we need our own doctors for 2 reasons. So one reason is that we don't want individuals taking this drug without supervision of a physician. We don't expect anything bad to happen, but you can't be sure until you test it. And so we need somebody who's aware of what's going on, and also who can be helpful in maybe measuring any of the changes, because we want to make sure that what we don't see is all placebo effect. We're not doing this as a controlled randomized clinical trial or crossover or anything else. We know that we didn't have a placebo effect in the child with Furry syndrome because she wasn't old enough to report on herself, and she did her own crossover study because she ended up off the drug and went back to baseline. So unfortunately, she was her own control. But I think both from a safety standpoint and an efficacy standpoint, that's important. And the Denver clinic is helping to make it easy to get the drug. Anybody's physician can get the drug directly from TaylorMade if they want to hassle with filling out the paperwork and doing all the things, and if they're in the right state. So it can go either way. I just think it's probably easier to go through the clinic in Denver, but it doesn't matter. And if it's a really young child and they don't have a participating physician in the Denver clinic that's in your state, then we'll just make arrangements for the doctor to directly apply to TaylorMade, and there's just some forms to fill out. So it's not much of a hassle.
Yeah. And I think the other reason my assumption is the Denver clinic probably wouldn't write the script unless you had a doctor who was willing to offer supervision. And is that right?
Yes.
And as SRF, what I dream about is this drug getting into a kid, the kid seeing improved symptoms, suffering less. But then when that happens, we will have 2 major wins. First, we will see what happens when you increase Syngap in a Syngapian, which we still don't know. Second, we will have one more clinician out there who's like, whoa, this helped a kid who I thought was really sick and probably maybe was just going to be symptom treated. And now there's a way to help them. And sad but true, doctors have learned to not take to take everything parents say with like a bag of salt, but they listen to each other. So I think the time we spend getting physicians to think about this, engage it, and come to believe that there's ways to help our kids can really have a lot of returns when they go back to their peers and the stories come from them. So getting our physicians engaged, encouraging them to use this very safe drug to try to help our kids, and keeping them in the discussion can really benefit the whole community. And with that, we're over time. Thank you, Bruce. Thank you, Kathryn. If there's more questions, go for it. But otherwise, is it only available in the US? The answer is currently yes, Beata, I think. Is that right, Bruce?
Only available in the US as far as I know.
Right now. But
Yes.
I'm sure if there's a way to figure it out, the polls will do it. Awesome, guys. Thank you all for your time. Thank you, Bruce.
Take
Great
care.
work, Kathryn.